Congestive heart failure is a mechanical failure of the cardiac muscle. As the heart weakens, its ability to pump blood out of the chamber, the ejection fraction, drops to dangerous levels. For decades, the standard synthetic intervention has been digoxin, an inotrope designed to force the muscle to contract with greater power. But digoxin has a notoriously narrow therapeutic index. The window between a dose that helps the heart and a dose that causes severe toxicity is incredibly small. Seeking a safer alternative, the Indian Council of Medical Research conducted a double-blind clinical trial to test a specific botanical extract. The trial concluded that the extract matched digoxin's ability to improve the ejection fraction, but without any of the observed toxicity. This botanical matrix is not a generic folk remedy. It is a targeted chemical solution to a modern pharmacological paradox. The source of this extract is Terminalia arjuna, an evergreen tree native to the Indian subcontinent. It is a riparian species, thriving for millennia along the same riverbanks that supported the earliest civilizations. Its most distinct identifier is the thick, cream-white bark, which naturally peels away in layers to reveal the primary source of its active compounds. Over 3,000 years ago, the Atharva Veda classified this specific tree for cardiac care in Khanda 5, Sukta 5.5. The text gave it the designation Hridayasya Ku, translating to friend of the heart. This ancient classification recorded a clinical reality that modern laboratories are only now beginning to quantify. In the ICMR trial, Arjuna extract matched the efficacy of synthetic digoxin, raising the ejection fraction by an equal percentage. However, the data diverged on safety. While digoxin showed a clear spike in toxicity, the Arjuna group remained at 0. This raises a biochemical question. How can raw tree bark mimic the mechanics of a synthesized inotrope? The bark contains high concentrations of natural cardiac glycosides, specifically Arjunoside and Arjunololide. These specific glycosides bind directly to the cardiac muscle fibers, dynamically increasing the force and depth of each contraction. It is the same mechanical pathway utilized by pharmaceutical drugs, but delivered through a different molecular structure. Arjuna achieves the mechanical goal of modern cardiology without the toxic side effects of isolated synthetic compounds. We can map these modern molecular actions directly to the symptomatic vocabulary found in the Ashtanga Hridayam. The ancient practitioner Vagbhata prescribed Arjuna not for general wellness, but for specific cardiac symptoms. He used it to treat hridspandana, the diagnostic term for palpitations and arrhythmia. He also identified its use for hridshula, the specific ancient term for the ischemic pain of angina. These practitioners were tracking and treating the same pharmacodynamics we measure today, relying on precise symptomatic observation. Unlike a single-target synthetic drug, Arjuna exhibits pleiotropic effects, meaning its botanical matrix influences multiple interconnected pathways at once. It functions as a natural ACE inhibitor, dilating blood vessels to reduce systemic hypertension. This vasodilation is triggered by stimulating the production of nitric oxide within the vessel lining, which safely lowers the internal pressure. Arjuna also inhibits HMG-CoA reductase, the same mechanism used by modern statins to manage cholesterol. Clinical data shows an 11 to 12% reduction in circulating LDL cholesterol. Simultaneously, the good HDL cholesterol rises by an equal 11%. By combining an inotrope, an ACE inhibitor, and a statin effect into a single organic matrix, the plant addresses the entire cardiovascular system. For these molecules to be effective, they have to reach the bloodstream. The ancient solution was a specific chemical extraction protocol. The Charaka Samhita details a method called Kshirapak. The protocol involves boiling the bark powder specifically in a mixture of milk and ghee. This is a deliberate dual-solvent extraction. Arjuna contains water-soluble tannins and fat-soluble glycosides. The water in the milk captures one set of compounds, while the fats in the ghee bind to the others, maximizing the bioavailability of the entire matrix. These recipes were not culinary traditions, but functional protocols designed to optimize drug delivery. This evidence spans 3,000 years. It began with the Atharva Veda's classification as a friend of the heart. It progressed into pharmacokinetic protocols found in the Charaka Samhita. And it leads directly to modern clinical trials confirming the plant's efficacy in treating heart failure today. This consistency is why traditional botanical medicine is increasingly integrated into global health standards. Because these compounds are powerful, any botanical intervention for heart disease must be conducted under the supervision of a qualified cardiologist. Integrative medicine treats ancient medical literature as a repository of empirical data, clinical observations that modern biochemistry is now quantifying and standardizing for the modern era.